Hello students, let us start with the next chapter in which we will learn about the structure of chromosomes followed by types of cell division. Before we could learn about the chromosomes, children, let us quickly recall whatever you have learned in your earlier classes about the cell, especially the nucleus. Now, we know that especially the animal cell has a outermost layer known as a cell membrane, in which we find one of the largest cell organelle, and that largest cell organelle is either it is oval in shape or spherical in its shape, and that organelle is nucleus, which is covered by a membrane known as a nuclear membrane, which has a perforations. And within this nuclear membrane we find the highly condensed network like structure, and that network like structure is known as a chromatin fibers. And within the nucleus we find the nucleolus, a spherical body found within the nucleus, and the nucleus encloses the nucleoplasm. And we know that outside the nuclear membrane we find a jelly-like substance, and that jelly-like substance is known as cytoplasm. And in the cytoplasm we find a number of cell organelles, right? Such as we find the mitochondria, in which the cellular respiration occurs, liberating energy in the form of ATP. And then from the nuclear membrane till the cell membrane we find the pathway for the movement of food substance, and that tubular structure is known as a endoplasmic reticulum. Endoplasmic reticulum, which comprises the spherical body on its surface known as ribosomes. Fine. So along with that we may also find the vacuoles, which are smaller in size. We find the lysosomes and many other cell organelles. Fine, children. When you look at the nucleus, very close to the nucleus we find one more cell organelle, that is centrosome. We find the centrosome. So what is the location of a centrosome? We find the centrosome near to the nucleus in the animal cell, right? And the centrosome comprises two spherical bodies, and those two spherical bodies are known as centrioles, and which are surrounded by the ray-like structures, and those ray-like structures are known as a aster rays. They are known as the aster rays, which help in the formation of spindle fibers, which are required during the cell division. Right? So in the animal cell, we find the centrosome situated very close to the nucleus, comprising two spherical bodies known as the centrioles, which is surrounded by the ray-like structures known as a aster rays. Fine. So let me show you the centrioles. In fact, the centrioles are at right angles to each other. They are at right angles to each other. Right? So these two are the bodies known as a centrioles, and these centrioles are surrounded by the ray-like structures known as a aster rays, and these structures are known as a centrioles, and the entire cell organelle is known as a centrosome. In fact, children, the centrioles are at right angles to each other. In fact, you can say that both the structures are the cylindrical bodies. They are the cylindrical bodies which are at right angles to each other. So thus you can say that the centrosome comprises two structures known as the centrioles, surrounded by the ray-like structures known as the aster rays, which help in the formation of spindle fibers during cell division. Okay, then how about the plant cell? How about the plant cell? In case of a plant cell, you find all these structures. Along with these structures, we also find one more layer surrounding the cell membrane, that is cell wall. Fine. And then we don't find the centrosomes in case of a plant cells. They have a microtubules which are found in the cytoplasm. So when you draw the plant cell, you can label it as no centrioles, no aster rays. But along with the cell membrane, they have one more layer around the cell membrane known as a cell wall. Fine, children. So let us start with the structure of the chromosomes. So just now I said that within the nuclear membrane or within the nucleus, we have this highly coiled network, or you can say thread-like structures known as a chromatin fibers. But these chromatin fibers, during the cell division, the chromatin fibers, which are the long thread-like structures, will condense, and then when they condense, we find a structure, a highly condensed structure, which appears in this way. Fine. So this is the structure of a chromosome. This is one single chromosome. Right? And number of chromosomes varies from one organism to the other organism. Right? Now take an example. In case of onion, there are 16 chromosomes. In case of a lion, there are 38 chromosomes. In case of human beings, there are 46 chromosomes. So it means the number of chromosomes vary from one organism to the other organism, whether it is a plant or an animal. So this is one single chromosome. I said we find the chromatin fibers inside the nucleus. So you will have as many as chromatin fibers, which are equivalent to the number of chromosomes. So if it is a human cell, we find around 46 chromatin fibers, and all those chromatin fibers, which are the elongated thread-like structures, will condense. They condense and form a chromosome. Right? So what is the definition of a chromosome? Chromosomes are the highly condensed chromatin fibers. So how do we define the word chromosomes? Chromosomes are the highly condensed chromatin fibers. Now, each chromosome comprises a centrally placed spherical body or a constricted region, and this structure is known as a centromere. So what is a centromere? So centromere is a spherical body or a constricted region along the length of a chromosome, okay, and to this spherical structure the arm-like structures are attached. So what do we call these structures? So you can say that there are 4 arm-like structures. There are 4 arm-like structures which appears like an arm. So what do we call these structures? We call these structures as sister chromatids. So we name them as a sister chromatids. So each chromosome comprises 4 chromatids, okay, and chromatids are attached to the centromere at a particular point. Fine. Now, what is the function of centromere? In fact, this centromere is attached to the spindle fibers which are produced from the aster rays, and the centromere will split up. The centromere will split up and these sister chromatids are pulled apart. Okay, which I will teach you in detail when I take up the phases of cell division. Okay, so what is the function of centromere? Centromere splits during the cell division so that the sister chromatids get separated by the contraction of spindle fibers. Fine, children. So we call this as a sister chromatids. Imagine if there is one more chromosome. If there is one more chromosome which also has a centromere, right? Again we call this as a sister chromatids, again sister chromatids, but we name this chromatid of this chromosome and this chromatid of the other chromosome as a non-sister chromatids. We name it as a non-sister chromatids. Then, why the name has been given as a chromosomes? In fact, chromo, the meaning of a chromo is color. Some is a body. It means it is a colored body. In fact, when you observe any animal cell or a plant cell under a microscope, you cannot identify the nucleus or a chromatin network or a cell membrane clearly under a microscope. So, in order to observe these cells under a microscope, there are certain dyes used in the lab. One such dye is the eosin solution. So, when we add eosin solution to the cells, especially the chromatin network, that is the chromatin fibers or the chromosomes, will absorb that color, and after which, if you observe those cells under a microscope, you can clearly observe these bodies which have taken up the color. Hence, they have been named as a chromosomes, that is colored bodies, because they are the ones which take up the stain of the dye that we add to the cells. Fine, children. So, thus you can say that each chromosome has a centromere at particular point along its length, and the centromere is attached to 4 chromatids, which are the arm-like structures. Fine? Yeah. Let us move on to the structure of the chromosomes we have learned. Now, these chromatin fibers or a chromatin network comprises 2 parts. It comprises 2 parts, which are the highly condensed network-like structure. It comprises 2 parts, and the first part is known as a DNA, and the second part is known as a histones. So, about 40% of chromatin fibers is made up of DNA, and remaining 60% of chromatin fibers is made up of histones. Now, histones are nothing but the spherical bodies. They are the types of the proteins found in the chromatin fibers. So, what are histones? Histones are nothing but the proteins found in the chromatin fibers which appear like a spherical bodies. Then, how about the DNA? In fact, DNA is a structure made up of 2 strands. So this is the first strand and this is the second strand. So DNA is nothing but a structure made up of 2 strands which is twisted, which can be compared to a ladder, with the steps, and this ladder is actually twisted. It is twisted, or you can say that DNA is a spiral or a twisted ladder-like structure made up of 2 strands. Fine. Now, how the DNA and histones are arranged? I said DNA is a structure made up of 2 strands, and we find these histones which are the spherical bodies. In fact, we have around 8 histone molecules. 8 histone molecules which are wound by the DNA strands. You can say that, okay. If you just look at this ball, you can compare this ball to a histone, and this ball, when it is wound by the wire-like structure, so this wire can be compared to the DNA strand and histones can be compared to the ball. Is it clear? So you can say that this is one of the strand. This is the second strand of a DNA. So like that you have around the DNA strand wound around around 8 histone molecules. So you can just imagine that there are around 8 histone molecules similar to these balls. Now when the 8 histone molecules are wound by the DNA strand, we call that structure as a nucleosome. So what is a nucleosome? It is a complex structure in which the 8 histone molecules are wound by the DNA strands. Fine. So a single chromosome may have millions of nucleosomes. I'm repeating, each chromosome may have around millions of nucleosome. So how do we define the word nucleosome? Nucleosome is nothing but a complex structure, or it's an arrangement, okay, a complex structure in which the 8 histone molecules or 8 histones are wound by DNA strands. Fine, children. Now let us study about the structure of DNA. So let us start about the structure of DNA. Already just now I told you that DNA is a structure which can be compared to the ladder with the steps. So this shape was first discovered by Rosalind Franklin, and later on the structure was discovered by two co-workers, that is Watson and Crick. So these are the two great scientists who discovered the structure of DNA. Yes, children. So let us study with the study the structure of a DNA. Yes. Now the DNA comprises two strands. So children, here you can see the two strands. The first strand followed by second strand. Okay. So when you twist this strand, when you just slightly twist this strand, when you slightly twist this strand, you have a structure like this. You have a structure like this, which can be compared to the double helix model of the DNA. Imagine in certain houses you have that spiral staircase. The spiral staircase which can be compared to the DNA strand. Right? So it means when you observe the diagram, it looks as though both the strands are meeting, but they are not meeting. They are totally twisted. Fine, children. So let us study about the structure of the DNA. The DNA comprises a number of repeating units. The DNA comprises a number of repeating units. In the sense, in one particular strand, okay, or in the other strand, you can say that DNA is a macromolecule or a giant molecule which has a repeated units in each strand. So there are a number of repeating units in each strand, and those repeating units we name it as a nucleotides. So here in the diagram you can say that this is the first nucleotide, second one, third one, or a fourth one. So these repeating units in each DNA strand is known as a nucleotide. So what are nucleotides? Nucleotides are the repeating repeating units in the strand of DNA. Or you can say the DNA strand is a polymer, the nucleotides are the monomers, the smaller units. And each nucleotide comprises 3 parts. It comprises 3 parts. What are they? The first part is known as a pentose sugar. The first part is known as a pentose sugar, which can be represented like a pentagon. And the second part of a nucleotide comprises a phosphate which can be represented using a circle. And the third one comprises the nitrogenous base. I said one of the nitrogenous base, right? So in fact there are 4 different nitrogenous bases. They are adenine, thymine, guanine, followed by cytosine. So what are the 4 nitrogenous bases? Adenine, thymine, guanine, cytosine. Right, children? So these are the 4 different types of a nitrogenous base. I said in one single nucleotide there may be one of the nitrogenous base. It can be adenine, thymine, guanine, or a cytosine. So thus you can say that each nucleotide comprises 3 parts known as a pentose sugar, which is represented like a pentagon, followed by phosphate, and then the nitrogenous base, which is one among these 4. So in the sense, you can say that here within this small space of a nucleotide, we find a pentose sugar. You find a pentose sugar, right? We find a phosphate radical. So this is a pentose sugar. So you may find a pentose sugar here also. In the opposite direction, you may find the pentose sugar. Then you will find a phosphate radical. So this can be the phosphate radical, which is represented like a circle. Fine. And to this pentose sugar, you may have a nitrogenous base which is arranged inwardly. So in the sense, the pentose sugar and the phosphate are arranged lengthwise. So again you will find one more pentose sugar here, one more phosphate. So one more pentose sugar, one more phosphate, which are arranged lengthwise, and internally you have the nitrogenous base for this particular strand. In the same way, even in the other strand you may have one pentose sugar and a phosphate radical here, again a nitrogenous base. So you can say that the nitrogenous bases are arranged inwardly and pentose sugar and phosphate are arranged lengthwise in each strand. Fine, children. So you can say that the steps of the ladder can be compared to these nitrogenous bases which make up the steps, and these nitrogenous bases which appear like steps we call them as a rungs. So rungs are nothing but the step-like structure of the DNA, which is made up of nitrogenous bases. Fine, children. Now, how are these nitrogenous bases arranged? How are these nitrogenous bases arranged? Quickly, let me draw. So this is one pentose sugar, one phosphate radical here. Then you have one nitrogenous base in one particular strand. Again you have one more nitrogenous base on the other, followed by one more pentose sugar and one phosphate radical. Right? Moving on to one more phosphate radical, then pentose sugar, then you have one nitrogenous base, one more nitrogenous base, a pentose sugar which is attached to the phosphate, and one more phosphate on the other side. So you can say that this is the first strand of DNA, the second strand of DNA, and you can say that this is the phosphate radical, pentose sugar and nitrogenous base, which makes the one nucleotide. So you can say that in the first strand there are two nucleotides, and in the second strand you have two more nucleotides. So you can say that the pentose sugar and the phosphate radical are arranged lengthwise in both the strands, and nitrogenous bases are arranged inwardly like a ladder steps. What do we call it as a rungs? And children, the arrangement of nitrogenous bases are always complementary. What do you mean by that? We find always adenine along with the thymine. So you can call it as a complementary pair. And between adenine and thymine, there is a weak double bond hydrogen, or a weak double bonded hydrogen bond we find between the adenine and thymine. Okay, and moving on to the next one, guanine followed by cytosine, which is one more complementary pair. In between the guanine and cytosine we have a triple hydrogen bond. So they are always complementary to each other. What do you mean by that? In case if you have a adenine here, what is the complementary pair for adenine? It should be thymine. Fine, children. If you have a thymine here, the complementary pair should be adenine. So you will never find the cytosine or a guanine with thymine. So always adenine is with thymine. If you have a cytosine here, you have a guanine here. If you have a guanine here, you have a cytosine. So hence, when you just look at each strand, if one strand has a adenine, definitely the other strand has a thymine as a nitrogenous base. If one strand has a cytosine, the other strand has a guanine. Hence we call them as a complementary pairs. Okay? Yes. Then what do you mean by genes? We have been learning this word. Genes are found in the chromatin fibers which determine the body feature of an organism, which is transmitted from parents to the offsprings. Right, children? Then what are the genes? Now just let us take an example of one particular structure. So just now I told you about the structure of a chromosome. A chromosome comprises centromere to which the two sister chromatids are attached. There are 4 chromatids attached to the centromere. Fine, children. Now, just let us take an example. What do you mean by genes? Right? Now, so these are the 2 strands of DNA. So you have a adenine here, thymine here, guanine here, cytosine here, again thymine here, adenine here, cytosine, guanine, like this, you have an arrangement of a nucleotides. So a proper sequence or a particular sequence of nucleotide will determine your body feature. So whatever you have received from your parent, so that arrangement of a nucleotides will determine your body feature. What do you mean by that? It can be the shape of your nose, the color of your skin, your blood group, okay, some people suffer from color blindness, some, where they cannot identify the 2 colors, right? That is a red and green. So they may have one particular set of nucleotides. So this set of nucleotides, the arrangement of this adenine, thymine, cytosine and guanine, okay, in that particular person will determine a feature in his body. It can be any feature. Maybe the thickness of your eyebrows. And there are certain cases where some people can roll their tongue, some cannot. So all these are the features of one particular person. And those features are because of this set of nucleotides. Fine, children. So how do we define the word genes? Genes are nothing but a sequence of nucleotides on a chromosome, a sequence of nucleotides. So how do we define the word genes? You can say that sequence of nucleotides on a chromosome that encode particular protein encode a particular proteins which express in the form of particular feature in the body. So just now I said the sequence of the nucleotides will determine a particular feature. So how do we define the gene? The sequence of nucleotides on a chromosome that encode a particular protein. So this determines one set of proteins which is expressed. So that set of proteins or that set of nucleotides will express in the form of a particular feature in the body. It can be anything that we find in your body, any body feature. Okay, now let us quickly learn about duplication of DNA. Duplication of DNA. What do you mean by duplication of DNA? Yes, children. So let us consider. Just observe a centromere. Observe this chromosome. So in this chromosome there is a centromere, and to this centromere you have 2 chromatids attached. But these chromatids undergo a duplication, okay, which is required for the equal distribution of chromosomes in the newly formed cells after cell division. So there is a duplication of a DNA occurring. There is a replication of a DNA which is occurring. Okay, let us quickly learn about the duplication of DNA. Okay, now during the duplication of DNA, these two DNA strands, so you can say that this is the first DNA strand and this is the second DNA strand, okay, which opens up. They separate, they open up, okay. So when they open up, okay, so this is one strand, the second strand, one strand and the second strand. Yes children, let me name it as a old DNA strand, old DNA strand and old DNA strand. Right? So these two strands, they separate. The DNA strands will separate because there are weak hydrogen bonds which makes them to separate easily. So when they open up, in case if you have a adenine here, you know that in the opposite strand you find the thymine. If you have a cytosine here, in the opposite strand you have a guanine. Or if you have a guanine here, opposite strand you have a cytosine. If you have a thymine here, opposite strand you have a adenine. So if this is the old strand, against the old strand you may have a formation of a new strand. So this is a new strand which is formed, and if there is a adenine here, definitely in a new strand you have a thymine formed. If you have a cytosine here, you have a guanine formed. In the same way, even on the other strand which has separated, opposite to that you have one more DNA strand formed. So if this is thymine, you have a adenine, you have a guanine here, there should be a cytosine. Children, observe carefully. Both the sister chromatids or both the chromatids are similar to each other, and hence we call them as a sister chromatids. Sister chromatids which are replica, which are similar to each other. And this formation of nucleotide or a new DNA strand continues lengthwise till the centromere. Even here it continues. So the complementary pairs are formed. It continues till the centromere, and now this chromosome having two chromatids, after the duplication, after the replication, instead of having two chromatids you have a four chromatids, four arm-like structures, okay, sister chromatids which are a replica to each other. So thus the duplication of DNA occurs, and this duplication is required for the cell division. Fine children. So in the next video we will start learning about the cell division, that is the types of cell division. Thank you.